Hi, welcome you all to Baggy Infotech Media. Greetings everyone. Today I'll be delving into the construction and screening of genomic DNA library during this discussion. What is meant by a gene library? A gene library comprises a compilation of gene clones encapsulating the genetic blueprint of an organism. Various types of gene libraries exist encompassing cDNA libraries, genomic libraries, and randomized mutant libraries. The applications of these libraries depend on the source of the original DNA fragments. The gene frequency in the genomic DNA library depends on the gene and genome size, as well as the copy number. The source material for the library is nuclei. These genomic DNA libraries are used to study gene organization and structure. Cytoplasmic RNA is the source material in case of cDNA libraries, and the frequency depends on RNA abundance and stability. The usage refers to studies that are related to the coding regions and gene expression. As we saw in the previous slide about genomic DNA library, the initial material, that means the genomic DNA, was cut into fragments with specific cutting sites using restriction enzymes. These fragments were then cloned into a suitable vector, amplified, and transformed into E. coli cells. Finally, the clones were screened to identify the desired ones. For constructing a cDNA library, first the mRNAs are copied into cDNA and then constructed into either a plasmid or a PAJ vector. After this, the final transformation to E. coli cells is performed and the clones are screened. These are the two major differences between the genomic DNA and the cDNA library construction. The basic material differ. The recombinant clones may represent the entire genome or the recombinant DNA molecules. This contains coding regions, introns, repetitive and non-functional DNA, as well as sequences that play a role in gene regulation. These techniques are utilized to examine chromosome structure or clone specific genes. Let me explain the procedures involved in constructing and screening genomic DNA libraries. As a first step, we have to purify and isolate the DNA. Then we have to analyze the DNA by the way of using agarose gel electrophoresis. Then the purified DNA may be digested using restriction endonuclease enzymes into small fragments, and that smaller fragments that can be cloned in plasmid or other suitable vectors. Cloning into suitable vectors depends upon the size of the insert DNA. So we have to choose the vector in order to the size of the DNA fragment you want to clone. Usually it is done by T4 DNA ligase, the molecular glue in a plasmid vector, and if the size of the fragment is within 20 kilobase pairs. The next step involved is the transformation. Here, the cloned plasmids will be transformed into E. coli. Transformation is a natural process in which cells take up foreign DNA fragments. These fragments are integrated into the host cell's DNA through non-reciprocal recombination. If the foreign DNA is not integrated, it will be disintegrated instead. Usually, calcium chloride treated competent cells are transformed with the plasmid construct containing an antibiotic resistance gene. Hybrid colonies with recombinant plasmids are visible on the media after overnight incubation. The other ways of genetic transformation include methods such as particle bombardment, electroporation, and microinjection. After transformation cells, there may be two types of phenotypes. One may be the transformed cells and the other may be the non-transformed cells. The transformed cells can be chosen by directly selecting them in an antibiotic medium. The transformed cells containing the recombinant plasmid, which are resistant to growth in an antibiotic medium, while the non-transformed cells may not grow. Based on this resistance mechanism, hybrid colonies can be selected for further analysis. The other ways of selection of recombinants are blue-white selection, DNA hybridization, and immunological selection. Followings are the some of the references you can very well follow for further reading. And I also like to acknowledge Temple University, Bharatdasan University, and Indian Council of Medical Research. Thank you.